Hello everyone, my name is Geraldine. I'm a postdoc researcher at the University of Melbourne, and the research that I am presenting today focuses on the microbiome gut-brain axis in Huntington's disease, or HD. So HD is a neurodegenerative disease that first affects the motor system and then the psychiatric and cognitive system of the afflicted, which gradually worsens as the afflicted ages. We have previously found evidence of changes in the gut bacterial population, also known as the microbiome, in both HD mouse models and patients. Now, using metagenomics data from the mouse model, we aim to identify changes in the gut fungal population, also known as the gut microbiome. So we first collected feces from the HD mouse model and their normal litter mates at 5 time points for metagenomic sequencing, from early age at 4 to 12 weeks of age, corresponding to young and early disease stage. Taxonomic assignment and quantification was done using Kaiju, and then using time-omics, linear mixed modeling splines were applied to smooth and model the temporal profiles, which were then clustered using sparse PCA according to their abundance profiles across time. We also used Mixomics R package for the multivariate analysis, as well as multi-omics integration. So, looking at the temporal profile of these fungal species at the first 12 weeks of life in these mice, sparse PCA clustering and LMMS modeling identified 6 fungal species that have different temporal profiles between the different mouse groups. If we compare the trend of the orange line, which is the normal group, with the HD group shown in blue line, we see that most of the differences are more evident towards the week 12 time point. Specifically, all of these were found to increase in the HD gut, with the exception of Penicillium digitatum at week 12. All the other fungal species would increase in the HD gut over time. The 2 key species that I want to highlight here are Candida glabrata and Candida paracilopsis. In the normal group, we see a decreasing trend of Candida glabrata over time and a steady trend of Candida paracilopsis in the wild type over time. And the overgrowth of these generally show no adverse outcomes in healthy mice, but has been shown to exacerbate existing gastroinflammation in mice. It was also reported in HD patients that the presence of Candida glabrata fungal structures were found in a specific brain region, which suggests that the infection by this fungus occurred at an earlier period long before death, as its secreted proteins were trapped during the formation of this brain region over the years. When we looked at the overall gut microbiome composition using principal components analysis, we found that there was distinct clustering of samples only at week 12, but not at any earlier time points. And interestingly, this time point coincides with the onset of the motor symptoms in these mice. So, delving further into week 12 time point, we aim to understand the potential inter-kingdom interactions between the gut bacteria and fungal communities. We performed supervised multi-omics integration using Diablo function from the Mixomics package. 7 species from each kingdom were identified to have high associations with each other, which are also distinctly differentially abundant between the normal and HD group. And what's particularly interesting is the association of a Lactobacillus reuteri with a number of fungal species, including Malassezia restricta, Glaria lozoyensis, and Rauwia lipolytica. In line with this, several studies have shown opposing relationship between Lactobacillus reuteri and fungal colonization, potentially due to the production of reuterin, which is an antimicrobial substance. So in summary, we found evidences of changes in the gut microbiome composition at week 12, which also coincides with the onset of motor symptoms. We also found increasing prevalence of several key species in the HD gut over time, and also our inter-kingdom integration analysis found Lactobacillus reuteri to negatively correlate with many key fungal taxa, suggesting alterations in inter-kingdom interactions within the HD gut. And with that, I'd like to thank all of those who are part of this work, and also thank you for your attention.